So, welcome everyone to this live stream today. Today we will be talking about repurposing drugs, and in particular, repurposing drugs from human to animal. And this is one of the topics that pretty much every pharma company would ask themselves at a certain point: should I do it? Should I, skip it? What are the things that I will have to keep into consideration if I want to do this? And today we will tackle some of the points that are interesting that we need to keep into consideration. So, without further ado, let's start with the presentation. And I will share my screen. Okay, wonderful. Let me get here in the bottom. So, before we start, a couple of words about us. At Level 4 Pulse, we are on a mission to have developed new therapeutics and new diagnostics for companion animals, especially, but for animals in general. And we are on a mission to help producing animal testing in animal and human health as well. And we do this with our little piece of work, which is providing biospecimens from animals. And here we talk about mainly dissociated tumor cells from cats and dogs, healthy cells from a wider variety of animals, frozen tissues and FFPE tissues, so formalin fixed, paraffin embedded tissues, especially from animals, blood and biofluids like blood, feces, urine, saliva, PBMCs, or other kind of isolated cells from the blood. And then on top of this, we have a portfolio, a complementary portfolio of antibodies, ELISAs, and growth factors, especially for animals. And then we also we collaborate with Altes Biosystem to provide a human gut model, a human replica to basically mimic the intestine. This is one of the building blocks, for example, for organ-on-a-chip systems that would also help reducing the use of animals in research. So, what is drug repurposing? And drug repurposing is the use of or the adaptation of existing medications, normally for human, to animals, to treat animals. And as you can imagine, this is not something that you can do one-to-one. There are quite some differences when we are developing, medicaments and going through the animal testing. There's always, this little voice in the back of our head saying, "Hey, we are not, 70 kilos of mice, so we need to be careful about how do we get our data, how do we interpret the data also produced with basically animal studies." The same obviously is true for the other way around. So if we want to adapt humans to human drugs to animals, we have to keep in mind that we are not, animals are not, or pets like a dog or a cat, they are not humans, but just, 10, 20, 30 kilos. Yeah? So it's just another different organism, and this individuality would need to be respected, and we will see now some of the aspects. But before we go into this, the question is, why is this even important? Why is this even a topic? And well, obviously, the market for pet medicine has been expanding quite rapidly in the past. And one of the reasons is obviously that the standard of care is higher compared to the past. So also pets would live longer, but also, from the perspective, like from a societal perspective, humans, they live longer. They would need also companion for longer time. So also the amount of animals, if we look to North America and Europe in particular, this is also growing over time. And so also the expenses, the expenditure in general for medicament, for veterinary medicament is increasing. So if we look at this graph here, we can see there's some data that we put together, looking at some of the latest publication on some drugs that have been tested and has been repurposed, and some other drugs that might be interesting and would probably be tested in the future. So it's a little bit of a mixed calculation. But then you can see in the dark bar, these are the actual sales figure for 2023 for human drugs. They're at the moment in the market. They're already sold in the market. And in the lighter bar, this is for the equivalent of the animal ones. Now they look quite different as these two bars, because obviously we have less animals than humans. I think there's approximately 200 to 300 million animals in both the US and Europe combined. But obviously this can vary a little bit. But one of the considerations that we have at the moment is also that because of various factors, one of those being also lower confidence in the efficacy of pet medicaments, just 50%, in fact, around 53%, this is one of the last statistics, 53% of the pet owners would bring their pet to the veterinarian when they are sick. And this is obviously quite surprising. Like if we look this in humans, I mean, not saying that 100% would go to the doctor, because probably, I mean, it's a lot of people would say it's not necessary, depending on how severe the situation is. But sometimes it's also about not being able to afford the medicament itself, depending, if it's particularly expensive. But this is definitely one of the data points that can and will improve in the future for pet drugs. And you can see already that the pet sales, they are forecasted to increase by 8.3% per year, and this is already 2 points more than the human one. So this is quite interesting data, that we might want to observe also in the future. And obviously, one of the main reasons for a drug company to consider repurposing, like a drug manufacturer to consider repurposing a drug, is that they've already done a lot of the work, a lot of the discovery work, at least. They probably already have some data in the different preclinical studies done with animals. So we just need to complement this a bit, at least on the safety aspect. They will obviously need to complement this also with efficacy data, with some more specific model cells and so on. There are specific of that particular animal model. And then they're basically good to go. I mean, they also have to run their clinical trials with animals. So, but most of the discovery work, from the perspective, is done. Now, this is not exactly the reality. This is not exactly what happens, and we will see now some of the aspects that will play a role into this. So, first of all, ADME tox. So ADME stands obviously for absorption, diffusion, metabolism, excretion, and toxicity. And these are some of the parameters that are used to determine basically the the pharmacology of a particular drug and what is the effect on the body, what is the the effect of the body on the medicament. And one of the first things that we can see is that there is quite some difference between different species. So whenever I'm producing a new drug, I would need to select the right animal model to test this in order to have, safety data that would make sense. The same way when I'm going the other way around. So here we can see this publication from 2022. And it's basically saying that in dog, the enzymes that are responsible for the clearance of orally administered drugs are cytochrome 3812 and cytochrome 3898 in liver and intestine. This in human, these are cytochrome 384 and 5, which are both in the liver and the intestine. So it's a quite different profile, and we need obviously to consider this to understand which one of the enzymes is responsible, what are the possible medicaments that are influencing with them, and potentially also slowing down or speeding up the whole process, the whole drug metabolism. A second aspect to consider is that drugs would have then different effects in different animals. So different animals would have different physiology, and so this will lead to a different pharmacology, and in general different effects. So here is an example from gabapentin. This is a drug that also in human is used for as pain medication and as an anti-seizure medicaments, and this is kind of similar with what has been used in dogs as an anti-seizure, anticonvulsant, and pain medication. But in cats, for example, this is used to treat fear and anxiety associated, especially with veterinary visits. So completely different. Completely not, but obviously that they are all acting on the central nervous system, but the effect in the end is completely different. A third point is a different dosage. And here we're talking about differences in the drug metabolism of the different animals, different speed, different clearance, and so then this is obviously, the other side is that this leads to a different dosage of the medicament itself. So we remain here with example of the gabapentin. For the dog is, for example, 50 milligram per kilogram, and for cats is 5 to 30 per milligram per kilogram. But then we can also see here, this is quite interesting. This has been taken from a meta review where they were looking at several publications, and there's not always really a lot of consensus about the doses, and this can also be because there's intra-species variability sometimes between different races of a particular or breeds of a particular animal species, but also different conditions would obviously influence this. So it's difficult to have a value that would be the same for everyone. Yeah, and this is obviously tells us that it is quite important to look how you translate the data that you created in these preclinical studies, in these animal studies, in the real world animal studies. So there's some difference. For example, I was talking in these days with a pharma company, human pharma company. They were developing, or they are developing drugs, and so the point they have to test it on different animals, and one of the animals for the gastrointestinal tract that is particularly similar to human is a dog, and they were quite transparent. They say, look, the main animal, the main dog, strain or whatever in in this kind of testing is the vivo. It's the only one that is used. It's easy to use and small, so you don't have to give huge doses or whatever. And so this means everything is related to the vivo. But there are quite some differences also in pharmacogenetic between different, animals. So you cannot translate the same exact, results to another, another animal. So you will need to another breed. So you will need to enlarge probably your study, have some more diversity into your study. So this is obviously one of the other difficulties. And then a fourth point is that there's a difference in the formulation. So you might have in your capsules, in your pills, in whatever, excipients, additive, things that would stabilize the formulation, keep it together, or whatever. And here you have to be really, really careful what you put inside, because what it works for a human, it can be really toxic also for animals. So here another example with gabapentin, they basically say, hey, the human oral solution contains xylitol, which should definitely be avoided for veterinary patients. So do not give your pet human gabapentin. I think this is also specified in on the package itself. And well, this is one of the things that needs to be kept into consideration. And a fifth point, and then we will stop here, is the different expression of the target. So this can also be really the groundwork that you lay the whole development on. This is what you really need to check. So it can be that the target is expressed at different levels in different tissues and different species, and so this leads obviously to a different effect. And here is an example of clopidogrel. So this is an antiplatelet medication that is used to prevent strokes and heart attacks. And normally it works by inhibiting B2Y12 with the receptor on the platelets. But this receptor is expressed at a much lower levels in cats and dogs. And so what basically, obviously, the effect of this medicament will be much reduced. And so we need to keep in mind all these kind of factors when developing a new medicament. Having said that, if you still have questions and or any doubt, or if you have maybe particular requests, or you want to investigate what are the possibilities to repurpose your drugs, maybe you're searching for biospecimens, and if you'd like to have an exchange, you can contact us either via Instagram, LinkedIn, maybe even YouTube, I think, or in the on the email that you can find here on my left side. So info@labforpose.com. You can call us or send us an email. Having said that, thank you very much, everyone, for being on the stream, and see you in the next one.